Why don't immune cells simply wipe out every tumor cell? Part of the answer: an immune cell carries both a gas pedal and a brake, and a tumor, to survive, presses the brake. This blue one is a brake the immune cell puts out. The paper calls it PVRIG. This gold one is a handle the tumor cell displays, called Nectin-2. Two proteins from two different cells, clasped head to head. This brake has two odd habits. It grips this handle tighter than every other receptor in its family. Yet at a near-identical look-alike handle, called Nectin-5, it won't even glance. Why so tight? And why so picky? The answer is hidden right here, in the hands they've locked together. First, how they clasp. The two faces fit together seamlessly, burying nearly 1,800 square angstroms, the largest contact patch in the whole family. What holds it so tight? Two keys. The brake extends an aromatic ring, phenylalanine F139, and the handle extends 1,2, phenylalanine F145, each plugging into the other's lock. One key each way, two locks in all. This is the paper's double lock-in key. Zoom in on the first lock. The handle's key, phenylalanine F145, buries itself in a hydrophobic pit on the brake's surface, wrapped all around by oily residues. This is the master key. Swap it for alanine, the smallest, plainest amino acid, and it's like filing the key's head flat. Binding drops straight to zero, no grip at all. Now the other side, the second lock. This time the brake extends the key, phenylalanine F139, into the handle's pit. Beside the lock sits a lid. A positively charged arginine, R95, presses right over the aromatic ring of a tyrosine, Y64, on the handle, and the pull between a positive charge and an aromatic ring clamps the key down harder still. But the brake's real trump card is a loop none of its relatives have, the CC' loop. It's longer than the others and warps entirely upward. The trick: a proline, P80, and proline loves to kink, folds the loop up, then its tip, an asparagine, an 81, flips over and throws out a whole string of hydrogen bonds, pinning the loop in its raised pose. Once raised, it grabs the handle in several extra places, and those extra grips are exactly why PVRIG holds on tighter than anyone. Keys alone aren't enough to keep it steady. Across the interface, there's a woven web of hydrogen bonds. The brake serine, S143, with just a small hydroxyl as the hub, reaches out like one hand pulling several of the handle's residues at once, and the bulkiest amino acid of all, a tryptophan, joins in too. 11 hydrogen bonds in all, stitching the two faces shut. Now the most decisive spot, the password that decides who is recognized. In the center of the interface, on a strand called the F strand, the brake puts out a positively charged leucine, K135, facing a negatively charged glutamate, E141, on the handle. Plus and minus, and measured right in the structure. Only 2.8 angstroms apart, all but touching, snapping shut into a charge clasp. This clasp both pushes the affinity to its peak and quietly decides which handle this brake will accept. So why not Nectin-5? Let's bring it in to compare. It isn't in this structure. It's overlaid from another one. In gray, it looks almost the same, lock and key all present. But at the very spot of that charge clasp, it swaps the charged glutamate, the charged hand that does the recognizing, for an uncharged leucine, which can't latch on. The most elegant proof is a swap experiment. Change the handle's glutamate to leucine, and the brake loses its grip at once. Flip it the other way. Change Nectin-5's leucine back to a charged glutamate, and the brake can hold on again. A single charge flips the entire choice. Back to the whole picture. On the axis where the handle is Nectin-2, PVRIG reigns alone. The other receptors can barely hold it. So if a drug could wedge between the brake and the handle and keep them from clasping, it would be like releasing the brake, letting the held-back immune cell go after the tumor again. That's exactly the logic of immune checkpoint therapy, the same playbook as the PD-1 antibodies you've heard of. Once you see that up-warped loop, those two keys, and that one charge clasp, you understand exactly where this new class of drugs takes aim.